Our next speaker is Zhang, who is a PhD student in our lab and is a PhD student at MIT, and who studies the function of link RNAs using CRISPR-Cas9.
Okay, hi everyone, hi again. So today I'd like to present some of our recent unpublished work on developing a genome-scale activation screen for studying long non-coding RNAs and then subsequent characterization of one of the top hits identified from our screen. The mammalian genomes contain thousands of loci that transcribe long non-coding RNAs, many of which have been shown to play very critical roles in different biological processes through a variety of different mechanisms. Some link RNAs, such as Tinker, for example, act non-locally or distal to its site of transcription by binding to target mRNAs and preventing their degradation. Tinker is very important during epidermal differentiation. On the other hand, link RNAs can also act locally. For example, the EXIST long non-coding RNA recruits histone modifiers to repress nearby genes and silence the entire X chromosome. More recently, the process of transcription of link RNAs has also been shown to be very important for local regulation of nearby genes. The process of transcribing the upper hand long non-coding RNA has been shown to keep the chromatin open for two enhancers shown in green and yellow for the hand-2 transcription factor, which is very important for heart development. However, even though there has been thousands of annotated long non-coding RNAs, most of the long non-coding RNAs that have been annotated have not yet been characterized. And the challenge relies on identifying functional link RNA loci and then distinguish between the many different mechanisms by which link RNAs can function. And so to address this challenge, we developed a genome-scale activation screen for studying link RNA loci. We designed a library of sgRNAs targeting over 10,000 different link RNAs and packaged it into lentivirus, then transduced it into A375 cells. A375 cells are BRAF V600E mutant cells. The V600E BRAF mutation is found in many different melanomas and is sensitive to the BRAF inhibitor vemurafenib. However, resistance quickly develops, so in our lab we sought to understand the mechanisms behind this resistance. So when we treat these cells with vemurafenib or control for 2 weeks, we can enrich for sgRNAs that target link RNAs whose activation then confers resistance to the drug. These sgRNAs will become amplified in the positive selection screen, and at the end of the screen, when we deep sequence the sgRNAs in the population of cells, we can identify candidate link RNAs that had been enriched and are likely contributing to BRAF inhibitor resistance. So at the end of the screen, we had a list of candidate link RNAs. And then to validate whether or not our screen worked, we individually expressed each sgRNA in the A375 cell line. And in the top panel here, I'm showing one of our candidate link RNAs. And in the top panel, we are showing the normalized percent survival in the presence of vemurafenib treatment. So each guide RNA confers significant resistance to vemurafenib by upregulating the candidate gene. And we also verified that the link RNA was upregulated in the bottom panel shown here. And we performed this validation for 6 different link RNAs. You'll notice that all of the link RNAs have interesting numbers, and that's because none of these link RNAs have been previously characterized. We then performed RNA sequencing on each of these candidate link RNAs to show that they have gene expression signatures that are consistent with vemurafenib resistance. And then we subsequently focused on one of these link RNAs based on our RNA sequencing results. This particular link RNA is located next to 4 different protein coding genes. And because we were the first to characterize this link RNA, we decided to name it Emissary. The reason why we thought Emissary was particularly interesting to study is because when we target the Emissary promoter with the Cas9 activator, we find that it induced dosage-dependent upregulation of 4 neighboring protein coding genes. So we wanted to explore this locus a little bit more. Now, We also found that this particular property was unique to the emissary promoter because when we target the Cas9 activator to each of these protein coding gene promoters, we did not observe the same dosage-dependent activation effect. Now, because there are 4 different protein coding genes that are upregulated and the link RNA was also activated, the next question is which of these components contributed to the screening phenotype, which is verapamil resistance. So to explore this further, we overexpressed each of the protein coding genes as well as the link RNA transcripts using randomly integrated lentivirus and found that only MOB3B conferred significant resistance to verapamil. What this shows is that the emissary does not contribute to resistance through a non-local mechanism. By overexpressing the emissary transcripts outside of this locus, they don't do anything towards resistance. And in fact, activation of emissary confers resistance through MOB3B. MOB3B itself is a novel kinase activator that we found mediates resistance through the Hippo signaling pathway. So the next question then is activation of emissary required for MOB3B activation and the subsequent resistance phenotype? When we targeted this locus, we were targeting upstream of the emissary transcriptional start site, but because emissary and MOB3B are bidirectional transcripts that share the same promoter, theoretically, if we target this region, we can produce resistance by simply directly activating MOB3B. So we really wanted to know whether or not emissary transcript is involved in this process at all. So we performed a series of experiments to test this out. We activated emissary with our Cas9 activator. Then we simultaneously knocked down emissary either through dead Cas9 targeting, polyadenylation signal insertion, antisense oligo targeting, and then observed the effects on MOB3B. Theoretically, if emissary is not important in this process at all, we will not see any effects on MOB3B because the Cas9 activator can just directly activate MOB3B and produce the phenotype that we want. So starting with the first emissary perturbation of dead Cas9 targeting, we engineered a bimodal perturbation system where we expressed dead Cas9 and MS2 and the activation components in A375. Then we introduced activation of sgRNAs with the MS2 hairpin that can then recruit the activation domains, or simultaneously repression sgRNAs without the hairpins that only recruit the dead Cas9 complex. So when we target the repression sgRNAs downstream of the transcriptional start site of emissary, we get repression of the emissary transcript. So by combining different activation and repression sgRNAs, we were able to show that expression of emissary and MOB3B are extremely tightly coupled across several different orders of magnitude. The next perturbation experiment that we did was we inserted a series of polyadenylation signals downstream of emissary transcriptional start site. And we did this using Cas9 mediated homology-directed repair. We generated 3 different clones with the polyadenylation signal insertion. What this does is it terminates the emissary transcript, so we're eliminating the emissary RNA without perturbing any enhancer elements in the DNA too much. So when we generated clones, the polyadenylation signal clones, we first verified that when we activated this locus, and the emissary transcript was knocked down in the poly-A clones. We then measured MOB3B expression and found that MOB3B was also knocked down in these clones. And because MOB3B is a protein coding gene that confers our phenotype, we also observed a downregulation in the verapamil resistance conferred by activating emissary. So these experiments provided genetic evidence that transcription of emissary is involved in MOB3B activation. And finally, the last emissary perturbation experiment that we did was we targeted antisense oligos to emissary to knock down emissary transcript. And again, we verified that emissary transcription was reduced upon upregulation when we targeted it with antisense oligos. MOB3B was also reduced in the presence of antisense oligo targeting to emissary. And so together, these 3 different perturbation experiments show that emissary transcription or the transcript itself is required for MOM3B activation. So this brings me to the next question, which is, well, which mechanism is it? Is it the process of transcription, or is emissary transcript itself recruiting a histone modifier, for example, that then modulates expression of MOM3B? So we hypothesized that if the process of transcription of emissary is important, then, because emissary shares a promoter with MOM3B, if we knock down MOM3B, we should also observe the same effect. So then we did some MOM3B perturbation experiments. So if we now knock down, using antisense oligos, the MOM3B overexpression, and then measured emissary expression, we also observed a corresponding knockdown in emissary. What these results show is that the transcription of emissary is important for the transcription of MOM3B, but the transcription of MOM3B is also important for the transcription of emissary. So together, the transcription of both the link RNA and the mRNA creates a positive feedback mechanism that then regulates the other 3 neighboring genes in that gene neighborhood. So in summary, we have developed a genome-scale activation screen to systematically characterize long non-coding RNA loci. And we've also used one of the candidate link RNAs to provide a framework for characterizing link RNA loci identified from these screens. And the link RNA loci that we characterize involves a novel link RNA and mRNA positive feedback network that then regulates a broader gene neighborhood that contains multiple genes, one of which is important for our screening phenotype. So in the future, we hope that this type of link RNA screen and subsequent characterization can help characterize the thousands of annotated link RNAs that are out there. And it also helps ask the question of what type of mechanisms are involved in link RNA gene regulation. It seems like from our screen, a lot of link RNAs regulate the expression of nearby genes, not necessarily by recruiting a protein that modifies the histone, but rather through the process of transcription or splicing, as many other recent work has shown. And finally, I'd like to thank all the people that helped me with this project. The project was really a collaboration between the Zhang lab and the Lander lab, and especially thanking Jesse Ungritz for helping out with the link RNA characterization. And with that, I'll take questions.
In this particular link RNA, I was kind of curious, along with the MOB3 and this complex of genes, just thinking about what its overall function might be. Question you could ask is pretty easy, is whether this is a highly conserved gene complex across mammals, vertebrates. Have you looked at that?
So it is actually quite difficult to look at link RNA conservation, but we know that the upstream enhancer that is of this link RNA is very highly conserved. The link RNA sequence itself does not appear to be highly conserved.
Can you hear me? Yeah.
Yeah.
So most people would say that insertion of a poly A signal actually would stabilize the RNA, so you're getting an opposite effect. Do you have any explanation as to why that's happening? Is it actually being polyadenylated? You're putting a poly A signal, but is it actually being polyadenylated?
So what we do to measure the transcript knockdown is we actually have a Tachman probe that is 500 base pairs downstream of the series of polyadenylation signal sites, and we do observe a knockdown in the transcript. But of course, we also measured the portion of the transcript that is before the polyadenylation signal insertion, and we did not observe a change in the upregulation of that portion of the transcript. So in our particular experiment, we did not see a stabilization effect.
What's the distance between the link RNA and the gene?
It's, so the transcriptional start sites are 66 base pairs apart. So it's very close.
Is it conceivable to actually knock out the link RNA and overexpress it on a completely on a plasmid or something like that to show that the cis function is required?
So in order to knock out the link RNA, we did that by using polyadenylation signal sequences, because that knocks out the RNA without having too much of, like, instead of deleting the link RNA, which can have a lot of effects on deleting like enhancers, for example, we did the polyadenylation signal sequence to knock out the link RNA, knock down the link RNA. And the rescue experiment that we did for this locus is we overexpressed MOB3B using an ORF plasmid, so outside of this locus.
So have you looked at the 3D structure of these 5 genes? Are they in a TAD domain or insulated neighborhood?
Yes, so we found that so we found that the link RNA is transcribed from the boundary of a topological TAD domain. And this particular TAD domain kind of encompasses the 4 neighboring genes. And if we look at the 7 other cell lines besides GM12878 that are in the Rao et al. Cell data set, we find that this TAD domain is conserved across all 8 cell types. So it's very conserved.
One last question.
C9orf72 is obviously a very important gene in
Yes.
ALS. Have you guys seen any mutations in that link RNA that appear to correlate with disease?
We have not yet had the chance to look, but yeah, it is certainly interesting that C9orf72 popped up as one of the neighboring genes. And yeah, could be very interesting to look at.
Thanks again, Julia.
Thank you very much.